Dear all, please make yourself comfortable, and we are ready to start our gene editing session at PMNET Forum 2024 here in Riga. And today I'm the chair of the session, and my name is Riekstina. I'm a tenured professor at the University of Latvia in precision medicine, together with Ulrika.
Yes, good morning. My name is Ulrika Beitnere, and I'm a senior scientist and project lead at the Max Delbruck Center in Berlin. It is my first time at the Precision Medicine Network and also my first time co-chairing a session. We heard yesterday that precision medicine has evolved far beyond genomics, embracing a wide spectrum of personalized healthcare strategies. However, gene editing tools are always evolving, and I'm very excited for today's talks, which will cover some of these newest tools that we can use for precision medicine.
And just to introduce some housekeeping rules, so we'll have our speakers giving their talk, then we allow one immediate question, and after all the talks, we invite speakers on the stage, and we have a panel discussion. So prepare your questions for the panel discussion. And it is my true pleasure to invite our first speaker, Pia Johansson, Director of Cell and Gene Therapy Core, Lund Stem Cell Center, Lund University. And I know Pia from Core EU STEM Network, where IPSC core facilities do work together. And I think Pia will give us latest flavor on her research on CRISPR and human iPSCs. So Pia, warm welcome.
So I would like, of course, to start with saying thank you very much to the organizers for a very nice event so far, and thank you for the invitation to speak here. So what I'm going to talk about today is maybe a little bit further away from like the precision medicine when it comes to cancer cells, but I think it is also, there is a role for iPSCs and gene editing inside the cancer field, and also these techniques, of course, applies to all cells in the end. So let's see. So iPSCs, though, is a really huge part of cell and gene therapies, or ATMPs. And as we can see here on the left, there is a projection of cell and gene therapies, like how much the market value is going to be in the coming years. And as you can see, this is quite explosive. But if we look on the right here, I think maybe it's too little to see, but what this one is showing here is that a large proportion, which is this one here, the orange one, of clinical trials currently going on for cell and gene therapies are actually originating in academia, so non-industry. And this is much, much larger than it is for small molecules or more traditional type of medicines. So this really shows that both iPSCs and CRISPR are really part of the future, and the academic basic research that leads up to these things are really important to pay attention to and make sure that that is standardized and done in a way that makes the translation then as easy as possible. Since I am in Lund, I would just like to say an example of a cell and gene therapy that has come out of academia. So STEM-PD, the clinical trial is called, and is headed by Malin Palmer, who is a professor at Lund University, but also the project lead for this. And this is completely based in academia, in research, and they've had their first in human trials where they injected cells straight into the brain of Parkinson's disease patients. And actually the first patient, even though the clinical trial isn't completed, but he has been on the news and so, and saying that now he can smell and play golf and do sports. So this is all super exciting, of course, to have that happening close to where we are, but also showing exactly this idea of these things coming straight from academia. Now, interestingly, of course, this is actually an embryonic stem cell based therapy instead of induced pluripotent stem cells, because this, of course, started like almost before both of these ideas of CRISPR and iPSCs came up. So let's bring it back to iPSCs and CRISPR. So together, they really are a magic duo. So iPSC, so let me just take you back. So this is, of course, induced pluripotent stem cells that are derived from somatic adult cells, and they have the potential to form all the different germ layers, and they are the induced equivalent of embryonic stem cells, meaning that there are much fewer ethical issues as well as less of a supply issue. So then we have CRISPR, which is the other part of the magic duo, and also that has had a pretty explosive sort of wave from its discovery. It's a cheaper and simpler and faster gene editing tool than the ones that existed beforehand. We will hear of some of the other ones as well today, but it has made gene editing much, much faster. So iPSC really is a revolutionary tool. So the idea is that you can then take human cells and work with human cells for your research. They can be derived from skin or blood or urine, and then you turn them into these induced pluripotent stem cells. And that could be either like control humans, so patients that don't have any known diseases, or you can have patient cells where they have a known disease, and either with a known mutation or with an unknown etiology, but you know that there is something in this genome that causes the disease, both of which are super important for disease modeling and disease understanding. And then, so when you have your iPSCs, then you can actually differentiate them into any cell type in the body, which makes it, easy to work with human cells, as it was not prior to this, right? Of course, the advent of organoids have also really made a huge difference in human biomedical research, in that now you can use 3D models, different types of systems that was also not possible before. So all of this is moving it forward. And then after you differentiated the cells, they can also be used for cell therapy, so it sort of goes back, go back into the body. Then if you add CRISPR to this, then you can do even more, right? Then you can change the genome of the cells. See that I cannot move away at all here. So then you can change the cells and correct mutations or induce mutations and so on. And that could also, of course, be used for cell therapy. And there are different ways that you can use gene-edited iPSC-derived cells for gene therapy. So one would be where you have a healthy donor, and then when it's maybe more of a common disease, and you can then gene edit that to induce, for example, with a CAR T, and then turn them into T cells, and then you can store them. They're basically off-the-shelf products, and then it goes to very many different patients, but not the original donor. But you can also have personalized medicine when it comes to these gene-edited iPSC-derived cells. So I'm not going to go into which type of diseases this could be, but basically you then take from one patient, you make iPSCs, you can then edit them, you can differentiate them to replace the cells, and then you put it back into one particular patient. And this is, of course, if there is a mutation that is unusual, a disease that is unusual, you can do all these changes and then put it back into the patient. But let's go back now for research, because it's not just about the clinic. So when you have your iPSCs and your edited iPSCs and you've differentiated them, you can then use them for a lot of different things. So, of course, basic research, disease modeling. You can also do drug screening and toxicity tests. And you can also do multiomics and large data sets where you can take patient cells from lots and lots of different cohorts and sort of put them together and then do the experiments at the same time. So then you really have a controlled cohort. For example, if you look at, if you want to do some neural study with patients with autism, they're collected over a long period of time, then you make iPSCs and then you can do the experiment in one go, so you don't have batch differences. So that's also what's really interesting about these type of things. So let's go to editing. So editing, almost all editing, with, of course, there are some newer ways now to do it without the DNA break, but almost all editing is based on the double-stranded break. And to do that in iPSCs, you do it like you do anywhere else. So you induce the double-stranded break, and then when that has happened, the cell tries to repair the break in two different ways. One is what is called non-homologous end joining, so basically the cell just tries to put it back together. This method is a little bit error prone, so often what happens is that there are insertions or deletions at the site of the cut, which leads to gene disruption or even like losing certain parts of the genome. The cell can also repair the break with a repair template, which can happen naturally, but in the terms of gene editing, we supply that repair template, and then you can really alter the DNA in a very different way. And this, so the non-homologous end joining is used then to create knockouts, small ones, either just one gene, or you can actually cut out pretty large bits as well. We have recently cut out some transposable elements. We have actually cut out 300,000 base pairs, which we weren't sure that was going to happen, but it seemed like that worked reasonably well. What you also can do a lot is correct a mutation, or you can insert a mutation if you want to sort of do modeling of that particular mutation. You can tag a protein or create a gene reporter, or you can insert a new gene altogether, for example, like the CAR. So you can really alter the sequence of the DNA almost in any way you want, which is a little bit crazy, but still possible, actually. So when you bring it back to iPSCs are really excellent because they can become any cell type in the body, but they are also a bit special. So in order to be iPSCs, they have to have these characteristics. So they have to have the right morphology, the right karyotype, and they are a bit prone to instability. They have to be in the undifferentiated state, of course, and they have to be pluripotent in order to actually be able to do the things that we want them to do. However, they have a tendency to really spontaneously differentiate, and there's a drift in the pluripotency and genomic alterations. And then when you add gene editing to this mix, you also have to think about that you might have really large off-targets happening due to the cut, either this allelic dropout that people have been talking about recently, or small off-targets in other regions, and also the toxicity of the components that we're adding to the cell. So that's why when we, in my core, so I run a core facility that makes these cells for researchers. So we either make iPSCs from skin or blood, and we also do a lot of edits. So this is what we do most. We take other people's iPS lines, and then we make the edits that they want. And this is our editing workflow, and you don't have to look so closely at it, but it's more to show that all the pink boxes here is quality controls. So you can also see it's a pretty long workflow, but also that you really need to make sure that the cells are what we think they are at all steps during this procedure. So that takes a bit of time, and it's important that you maintain the quality. However, there are also other CRISPR tools, which we will hear more about later, but the idea is here that it's not just that Cas9 cuts the DNA. You can actually mutate the Cas9 so that it doesn't cut DNA anymore, but it still uses the same technology with this single guide RNA to guide it to a specific region of the DNA. And then you can fuse things to the dead Cas9, as you call it, if you have mutated these two nuclease domains. If you add a CRAB domain, then it becomes repressive. If you add an activator domain, then you, of course, get activation, if you put them at the right place. So this is CRISPR-I and CRISPR-A, respectively, here. And I'm going to show a little bit of example data here, because as you saw, the gene editing pipeline is quite long. You have to go clonal. Whereas if you want a gene knockout, CRISPR-I can actually be another alternative. So here is an example of where we use CRISPR-I, where we knocked out, or it turned out to be a knockout. So we use CRISPR-I to silence a gene called zinc-finger 558. And what we can see here, basically, is that we have very, very good knockdown. And if you look at the activation mark, if you look at cut and run for H3K4 3 methylation, as you can see over here, Here is the activation mark in the controls, and then that is completely gone after the knockdown. So this is basically a knockout that we created here. And this is, of course, a much easier procedure. We use the bulk here instead of going for clonal, which when you use viruses, of course, is actually beneficial. So this is basically creating a knockout in a different way. And this is very stable. We made organoids from these cells, and like 4 months after we had initially started the CRISPR-I, it's still expressed and still knocking down this gene or this protein. So it's a very efficient way to do things in a slightly different way. So, but when it comes to iPSCs, there are some like gene editing challenges as well, and that comes particularly when you're working with organoids or also other differentiation protocols, because we often do the gene editing inside the iPSCs, not in the cell type of interest, because it's often hard to reach high enough numbers of cells inside the organoids. So what we really need is inducible mutations. So you basically do the basic gene editing inside the iPSCs, you differentiate them, and then you turn on the edit, so to speak. And some of those things are not so hard to do. So you can do CRISPR-I, CRISPR-A with inducible constructs. So they're only turned on once you give doxycycline, for example. You can also do inducible knockouts in a very similar way to one does it in mouse genetics, which has been done for a long time. But then the question is, how do you induce a SNP? If you want to see it only once you come to the neural fate, for example, how do you induce it? And this I don't know, but I think that this is something that will come a little bit later to make it even more sophisticated, actually. And then I would like to also just make some advertisement for core facilities in general. And I think what we are is like we are hands and expertise for hire. These molecular tools are quite complex, and often you maybe just need to do it once. It will cost a lot of money and a lot of time to get the expertise into your lab. Instead, I think you should give us the opportunity to make your tools so you can then focus on the research questions. And of course, we try to think that we are keepers of quality, and we keep as standard operating procedures as possible, and we try to really make sure that we keep up to date what is required for publication, for maintaining the cells in the best possible state. So I'm here also, I spent a lot of time getting some AI to make this picture, but I was hoping that, that this conference will also start to do a collaboration between our two countries. Like our services are available for all academics, international and national, and all our products are very easy to freeze and transport. So I think we can really start a lot of different collaborations, or if you need help with setting up core facilities, then we can also help with that. So that is my last slide, and I would just like to thank you for listening, of course. This is the team that does everything, which are doing a great job. Here is the website that you can scan if you're interested, and these are our services. So as you can see, we make iPSCs, we do gene editing, but we also make viruses and mRNA at the moment. But feel free to contact us, of course, and yes, thank you for listening.
Thank you, Pia. That was a beautiful talk indeed. And if there's any immediate question, then please. Yeah, Ulrika has one.
I actually have one. I was curious, what kind of quality checks do you offer once you have done the gene editing at your core? Because I can imagine that there could be a lot of surprises. And what are the tests that you offer?
Yeah. So what we do, sort of part of the standard pipeline, is that we do a SNP array. So genetic instability is the biggest problem that we have, and that's often actually the clonal expansion that causes that instead of the gene editing itself. But so we always look for copy number variations all over the genome. They are sent to G-banding. We also check that they are still in their undifferentiated state. We do also offer off-target analysis if people want that, so top 5 sites. And then we also now started to do this very restricted copy number variation analysis, because it's also recently turned out that when you think something is homozygous, it's actually some of the time hemizygous, so the other part of the gene is just completely gone, but you don't see it because you use Sanger sequencing, and if it's disappeared, you think it's homozygous. So we just started to introduce that into our standard pipeline as well, to sort of just look for that there are 2 copies at least close to the cut.
Great, thank you so much.
Thanks again. Wonderful talk.
And with that, it is my great pleasure to introduce our next speaker, Dr. Giedrius Gasiunas. Dr. Gasiunas is a highly cited researcher in the field of CRISPR technology and one of the pioneering scientists in its development. For those who might not be from the field, the Lithuanian team, which includes Dr. Gasiunas, played a crucial role in the early work on CRISPR, nearly becoming the first to describe its mechanisms in 2012. So he's currently affiliated with Vilnius University and is the co-founder and chief scientific officer of Caszyme. And his talk today is titled Engineering Compact CRISPR Nucleases for Genome Editing. The stage is yours, please.
Thank you for kind introduction. And also, first of all, I also would like to thank organizers for inviting me to be here. And as was presented, I have also played some role at Vilnius University, but today I will mostly talk what we're doing at a company called Caszyme. And okay, so I'm trying to switch slides, but...
Please try to use maybe the remote.
Okay, it started to work. Okay, so basically what we do, since we have a long time experience in CRISPR, we try to use it and enable our partners to work in CRISPR field. And actually, I very liked analogy which mentioned by Pia, that actually I think we provide the heads and then hands for hire in CRISPR field. So who we are? So company was established in 2017. And main co-founder is Professor Giedrius, who was behind, one of the scientists behind the elucidation of mechanism of Cas9 nuclease, which later was one of the breakthrough of this field, and also was involved in this work because at the time I was a PhD student and postdoc in Professor Giedrius' lab. So we are located in Vilnius, Lithuania, so near from Riga. It's only 3 hours drive. And as a company, this year we grown up by incubator and then moved to new labs, our own space. So we have space to accommodate more projects. So of course, as you heard from first presentation, main application of CRISPR and Cas protein is genome editing. So, and to edit genomic DNA, you firstly need to introduce a double-stranded break in the exact position of genomic DNA. And then double-stranded break is repaired by the cell machinery, and then the side change is introduced. And it's difficult to overestimate the importance of this technology, because it's actually revolutionized research, because I think now it's used as a research tool in many labs, but also it's important in medicine. I think in these 2 days we had a lot about T cells, these CAR T cells, and also some trying to making cure for some genetic disorders. And also it's becoming more and more important in another field. It's agroscience for advanced plant breeding. So also some of you heard that actually now, even in Europe, with the European Parliament tries to introduce some changes in regulation of how in plant GMO plant regulation, and since genome editing can introduce changes which are very the same as can be acquired naturally, we hope that such plants will be not regulated. So, and Cas9, so as I mentioned, you need double-stranded DNA breaks to get gene editing as a first phase, and Cas9 was a very nice tool for that because it's has dual origin, so it's Cas nuclease leaves DNA, and guide DNA directs enzyme to cleave doubles to recognize the DNA. So this, as you know, it's quite easy to change nowadays RNA sequence. So just by therefore Cas9 can be guided to DNA sequence. It separates DNA strands. Structure called a loop is formed, and both DNA strands then is cleaved by Cas9 and double-stranded DNA introduced. And once such mechanism of Cas9 was demonstrated, I think many scientists show that actually Cas9 can work in eukaryotic cells and human cells and introduce changes. And I will not expand here, but as Pia already introduced, there is two major pathways how DNA is, how double-stranded DNA breaks are repaired in cells. One is called NHJ, which introduces some small deletions or insertions and called indels, and another HDR, which leads to large insertions or correction of the sequences. And the importance of this finding of Cas9 could be illustrated also that actually last year, only 11 years after this finding of initial mechanism of Cas9, first CRISPR medicine was approved, firstly by FDA in US, and actually it's mostly for genetic blood diseases like sickle cell disease, and it's ex vivo therapy. So cells are collected, edited, and then again injected for the patient. And this, I would say, it is simplest way to do CRISPR editing. But if we think about some in vivo editing, like editing some cells in adult organisms, everything goes much complicated. And even though Cas9 is quite very successfully used by many labs, it still has some limitations. One is that actually this not only guide RNA but also an additional sequence called PAM is required for target selection. So it means that you not always can target the sequence which you want. It's very important, for example, when you want to edit only one allele of two, or when you target SNPs with base editing, then you not always can achieve this target sequence you want. Also, for delivery in vivo, main mechanism is used adeno-associated viral vectors, and problem is that it has limited cargo size, and large proteins like Cas9 is Streptococcus pyogenes Cas9, which is most widely used now, do not fit into single viral particle. Also, another concern is safety, because sometimes Cas9 can cleave other sequences in genomic DNA, and another issue which is interested also in, especially important for companies is intellectual property, because at least in Cas9 field is yet complete mess with IP, and especially there is still fight in I think interference between two major patent holders in US, and in Europe, for example, also there's some. So could be different regulation, and it's also do not motivate companies. So therefore, we try to address some of that issues by searching of new tools. And how we do it, we coming back to fundamentals of CRISPR. And actually, this CRISPR originally was and there's immune system of bacteria cells, which protects bacteria from bacteriophages, and they are very widespread. So it many, like about half of bacteria species, and most of Archaea has its own CRISPR systems. And because viruses mutates very rapidly, CRISPR also changes very rapidly, and it's creates diversity of proteins. And actually, if you look at literature, you will find that actually now not only Cas9 protein is used for genome editing, but also there is another family called Cas12, which actually is very diverse from Cas9, because if you look at Cas9 proteins, which also are quite different in sequence, but they structurally and they are very similar and has the same domain organization, but Cas12s usually are quite distinct, and also only common thing between them is so-called Riekstina domain, which cleaves DNA. So today I will exemplify our work with two examples, two nucleases on which we are working, which belongs to Cas12 family, and they are smaller than Cas9. They recognize different sequence. They are different than Cas9, so addresses, at least from initial analysis, it looks that it addresses at least three drawbacks, could address at least 3 drawbacks of Cas9 technology. So I will start from introduction of enzyme called Cas12L. Cas12L actually stands for Lithuania, but it came up that actually there was a lot of Cas12s, and this letter was free to name. And actually it's interesting for this family, so we found it by searching in the bacterial genomic DNA for proteins which has Riekstina nuclease domain and are located near so-called CRISPR repeats. And actually, what was interesting that only 4 similar proteins were available in all public space, and also this protein was so different that except of Riekstina domain, we didn't manage to identify any other domain informatically, and even when AlphaFold was released, it could not predict the structure of Cas12L. So initially, we wanted to look if Cas12 cleaves DNA, is the same Cas nuclease. So we cloned this Cas12L system into E. coli vector, modified it by including new RNAs which target our design plasmid library, and which contains like randomized region at PAM sequence, because we still cannot predict the PAM sequence of enzyme from protein sequence. So by digesting this library, we capture cleaved products and then can determine PAM by illumina-deep sequencing. And what we showed that actually this enzyme can cleave double-strand DNA, that it was recognizing CCY PAM, so it's PAM is a little bit longer than Cas9, and also that it's different from Cas9, but similar to other Cas12s, cleaves DNA, leaving like 5 to 9 nucleotide overhangs. However, when we try to just cleave DNA, some DNA targets in vitro, in tube, we observe that there is like a very big difference between efficiency of cleaving targets, even in vitro, even though they had correct PAM. So what we try to do, we try to use, instead of linear DNA as a fragment, to use supercoiled DNA as a substrate. And in supercoiled DNA, strands are like twisted in different directions than DNA double-stranded DNA. Therefore, for nucleases, it's easier to open DNA and recognize and cleave DNA targets. And what happened, actually, these targets were cleaved in supercoiled DNA, were cleaved very efficiently. So we thought that most probably this enzyme should have some problems with binding of DNA sequences. And when we use the same best targets in human cells, we observe that it either do not have any activity or have very weak activity. So we needed somehow solve this binding issue and make protein working in human cells. And since AlphaFold didn't help, so we in collaboration with Vilnius University, we solved create structure of enzyme with guide RNA and DNA substrate. And actually, it was surprising that structure of enzyme also was quite different compared to other enzymes, and also it had very long and quite complicated guide RNA. So we use this structural data to introduce mutations which might increase binding capability of Cas enzyme, and actually by introducing some individual mutations, we observed that there is like mutations which increased enzyme activity in cells by 2 or 3 folds. And when we combined all the mutations, we demonstrated that actually combining of some mutations increased enzymes activity in human cells very substantially, and more than half targets now are working in human cells. So summarizing, with Cas12L, we now can target the same targets as with SpyCas9, because PAM is in similar location but in different strands, but different pattern of the NHEJ, because for Cas9 you usually get small indels as most mutations, and for Cas12L you get larger deletions. So summarizing, we identified Cas12L enzyme and showed that it can cleave, can be used as potent genome editing tools. Another story related to Cas12F enzyme, which actually is even shorter, about 500 amino acids. So in length, it's shorter, like much shorter than Cas9 protein, but it has also some drawbacks. So its function as a dimer, so it's more complicated mechanisms of cleavage, and also we observed that it has better activity and elevated temperatures, so about 50 degrees. So it means that it's like not so active at 37, which is needed for editing of human cells. So in this case, in collaboration with our partner Cortiva AgriScience, we designed yeast-based assay which relies on disruption of AD2 gene. So if AD2 gene is disrupted by Cas protein, the colonies get red, and actually initially we screen like wild-type protein. You see that we get red colonies only 45 degrees. Next, we made library in which each amino acid was changed to any other amino acid, and it contained and one variant contained only single mutation. And in this case, we started to see some pick up some colonies in 37 degrees. And when we combine these mutations, you see that activity was good in yeast even at 30 degrees. So when we purified proteins, actually we demonstrated that actually thermal activity of protein shifted to lower temperatures. But of course, made questions: does it translate well to activity in human cells? So actually, experiments in human cells also confirms that we see increase of 10, 15 times of activity in human targets. So summarizing my talks, I demonstrated that actually not only Cas9 could be used as genome editing tools, but also novel Cas proteins such as Cas12F or Cas12L. But of course, most of them still need some mutagenesis and optimization before using. So, and as I mentioned, we worked for this project with some partners like Corteva AgriScience and New England Biolabs, Vilnius University, but also we have some quite broad collaboration network, including Thermo Fisher Scientific. We had projects with Thermo Fisher and also other companies. And in the end, I would like to thank all the Caszyme team, which were involved in the project, also our collaborators from Corteva, New England Biolabs, and the Vilnius University. So thank you.
Any immediate questions from the audience? I have a quick question regarding the size. Since it's so important for delivery, and it's kind of known it's easier to deliver smaller nucleases, do you think we have already hit the limit of the smallest, still, let's say, specific nuclease that can be used?
Yeah, so actually in CRISPR field, yes, but there is also some other proteins like progenitors of Cas9 as Cas12L, which are usually located in transposons. So it's called 10PB SCB. So they might be a little bit even smaller, but still not much characterization is done yet, and I think it's not clear about activity.
Thank you very much.
It is my pleasure to introduce our next speaker, Ulrika Beitnere. She is a senior scientist at Max Delbruck Center for Molecular Medicine in Germany, and her current project is aimed to enhance the functionality of stem cell-derived organoids and she leveraged her CRISPR expertise for functional screening. Originally, Ulrika is from Latvia. She graduated University of Latvia with PhD in pharmacology, and then she went to California to Professor David Segal's lab, and she worked on gene editing as a gene therapy for Angelman syndrome. So this is going to be also her talk today. So welcome, Ulrika.
Thank you very much. Thanks for the kind invitation. I want to first start by expressing my sincere gratitude for being here. It's very special for me to present my postdoctoral work from the University of California, where I did my postdoc. And let's just move the slides. Okay, and just want to start with, yes, the work was not done at the Max Delbruck Center. It was conducted at the University of California, Davis, and the work was funded by the foundation of the Angelman syndrome Therapeutics. And I want to also give a big shout out that there is now also a special Angelman syndrome group in Latvia, which was formed just last year. So I just want to give a big shout out to them, since I'm also a very proud godmother of this this society. And so for those who are new to Angelman syndrome, I want to start with the story of a little girl named Quincy, who was born as the second daughter to Allison Barrett in New York City. Very early in the development, basically in the first couple of months, her parents noticed that she's not meeting a lot of the important milestones that they saw in their older daughter. And first the pediatrician said, please wait, everything will be fine. But the parents insisted that they wanted to have a genetic analysis that was showing that she has a rare neurogenetic or neurodevelopmental disorder called Angelman syndrome, which is approximately 1 in 15,000, but actually could be even more common. 1 in 8,000 are some of the numbers that some studies have shown. And it is a very severe mental and physical impairment. Quincy has difficulties with sleep, has also sometimes epileptic seizures, and has problems with walking. And as you see, in this picture with her siblings, she's using a special device to communicate with her parents because she's nonverbal. And also, CTI showed that she has a smaller brain compared to an age-matched child in this age. And also early on, there was noticeable that she has gastrointestinal problems, problems with digestion, constipations. And on that, we also studied that in Angelman syndrome animal models and show that even this one gene that is missing can affect also the gut system. But interestingly, like for Angelman syndrome, I want to also note that they have a very happy, excitable demeanor. They smile a lot, laugh, and have special like hand flapping movements that makes it kind of different from other like severe autism-like disorders. And interestingly, also for the genetics, it is a most often de novo, so a random thing that happens during early development. And so going back a little bit, stepping back a little bit, why this is a talk that I brought to you in a precision medicine meeting is that until a few years ago, the idea of curing genetic conditions such as Angelman syndrome seemed outlandish. It would even seem irresponsible to give like false hope for families. But here we are. Here's an example where a lot is known about the genetics that causes the disorder, and also a lot of work has been done identifying the key proteins, pathways involved in the pathogenesis. In this case, an enzyme, ubiquitin ligase. And that one piece that is missing is the clinical translation. And so the story is about hope because Alison Byrne, together with other Angelman syndrome parents, did the unthinkable. They raised millions and millions of dollars and brought not only like one research team, but 30 different research groups in the US and across the world who are working towards different kind of approaches how to solve this problem of the missing gene in Angelman syndrome. And so I was part of the group that worked towards the artificial transcription factor and zinc finger and also the CRISPR approach. But also there are multiple other groups who worked using the replacement therapy and also azos, the antisense oligonucleotides, and also the enzyme replacement therapy. So there are a lot of different ideas, and it is true that in a situation like this, and I will tell you a lot more about the genetics, multiple things could work, for some of the Angelman syndrome patients, because it is also a diverse group of people. And so now to the genetics. It is a very specific brain neuron, specific genomic imprinting disorder where the UB3A gene in our neurons in the brain is regulated so that only from the maternal allele it is expressed. So the paternal allele, the father's copy of this gene, is silenced with a long non-coding RNA strand. But what happens in Angelman syndrome is that we see a very large deletion, in some cases also mutations of the UB3A, which makes it non-functional. Also others have less common cases where it's uniparental disomy, so it's basically silenced on both alleles. And so this gives us different ideas, different approaches how to address this problem. And in the group where I did my postdoc, we focused on silencing the UB3A gene from the paternal allele, which can be done with multiple different genome or epigenome editing tools. And so just for those who are, again, not doing gene editing in their work, I just want to give you a little bit intro about the different editing tools besides CRISPR. Everyone knows about CRISPR, but before that, we know about zinc fingers, which are DNA binding proteins, and also transcription activator-like effectors, which are a bit different. You have to design the specific protein towards the site in the genome. And similarly, like with CRISPR, where you can fuse a repressor or activating sites, you can do that also to zinc fingers. And so about the work that I will present today is about the treatment using the artificial transcription factor, where we want to bring it in early at the site where the long non-coding RNA strand starts. And and we also did a lot of work on CRISPR-Cas13, but because of time, I will not talk about this today. So what we did with the zinc finger is that we designed One that is targeting the site in the human genome and also in the mouse, because we wanted to use the model that describes and shows some of the behavioral problems with locomotion. And so we were able to find a zinc finger which has the repressor crab domain fused to it that is successful in doing so. So you can see that the SNRF site, which is early for the long non-coding RNA strand, is really reduced when we use this therapeutic, which we called S1K, for the mouse, and in the human 555. And so using also Angelman iPSCs that were differentiated into neurons, we also saw that the UB3A antisense is going down, and also the UB3A gene is going up. But also important to note, because where we are taking this zinc finger in the genome, we are affecting these non-coding RNA strands, NORD116 and 115 as well. And I will briefly touch about what it means for the field that we are affecting these non-coding genes. So going forward, we did a study with an adeno-associated virus, because it is kind of the viral vector to use for brain-wide distribution. If you want to introduce your therapeutic to as many neurons as possible, you will use AV in animal models. And there are a lot of efforts going also towards translating that to humans and now non-human primates. And so what we did with one single injection, after 2 weeks, we did behavioral assays, and after a couple more, 2-3 weeks, then we analyzed the brain tissue from these animals. And indeed, we were able to see that we are bringing back the missing UB3A protein. And in a Western blot, here you can see the wild type has so much more UB3A that you had to normalize the levels that you put on the blot. But indeed, we are bringing back the UB3A to levels that are significant. And also here, we can see that the therapeutic is really delivered. And again, the SNRF and SNRPN site where we are targeting is downregulated with this treatment. And similarly, like we saw in the human neurons, we are affecting the SNORD116, 115, and also the UB3A antisense is significantly going down. Additionally, we had to analyze different sites in the brain to see how well we are delivering this AAV zinc finger. And here you can see that some of the injections actually were not as precise as we thought, and so we kind of divided the group into low concentration of the virus and high concentration, because we could really noticeably see that the more of the therapeutic, like in here, the S1K got into the brain, which you could see in this image, the more UB3A you have. And so less is maybe not efficient. So you have to achieve a significant amount of neurons to really see the effect, although even a smaller amount is efficient in the behavioral test that we did to see an improvement. And so then we also had to ask the question about specificity and tolerability, and so we did an RNA-seq bulk where, in this case, we were able to detect the S1K protein, I mean the gene in the mouse brain, but we were not able to see any immune responses, none of the inflammatory responses such as interleukins, IL-2, IL-15, IL-18, or tumor necrosis factor A or interferon G were higher expressed in these animals, which shows us that they were tolerating this treatment quite well. And last but not least, we also did the behavioral rescue tests where we were looking at the locomotion, how well these animals are moving around, because this model with the missing UB3 gene really is affecting the animals quite a lot. They are moving much less and have difficulties with their gait. And we were successfully able, so here in the blue you see the animal is the AS animal, and here with the treated one, the red one is much closer to the wild type. And similarly here, the red is closer to the wild type levels, and the total activity was definitely increased, which is really great. We were not able to show a rescue in Road, but the gait analysis showed improvements even in the gait, which is an important, again, translational readout for clinical trials. And so with that, I'm already at my summary slide, where I just want to show you that with this zinc finger that we targeted early on at the start of this UB3A antisense in the mouse and also in the human, we were able to detect and rescue molecular and behavioral phenotypes. And I kind of want to end this that this is just a small step towards a clinical trial, but the field is moving towards that we have already some of this therapeutics in clinical trials, and if there will be more and more success stories, and there are already some success stories in the Angelman field, then we will change the way that we think about genetic testing and autism. So it's a very exciting time to live in, and I want to thank my supervisor, David Siegel, and the whole group that I was part of, and I'm happy to take any questions. Thank you.
Thank you for a beautiful talk. Please, one question now, if there is from audience. If not, if I may ask, so you said you use AAV virus to target the central nervous system in mouse model. Was it a particular subtype of AAV that you used?
Yes, we used a special capsid that has been designed that can cross the blood-brain barrier. So that allows for systemic injections. You don't have to even invasively drill a hole in the skull and find the right spot in the brain, and that allows for a less invasive therapeutic. So that is also something that is in the works for translation, where we could find the right kind of virus capsid that can cross the blood-brain barrier also in humans.
Thank you. Very interesting talk. Okay, thanks very much.
Here I will introduce the next speaker, Dr. Anna Klucnika. It's with great pleasure. She is the platform leader for iPSCs and gene editing at Laverock Therapeutics, a UK-based biotech company. Laverock is pioneering the development of programmable next-generation cell therapies, and Anna is playing a key role in establishing the CRISPR-Cas genome editing workflows for induced pluripotent stem cells. The stage is yours.
Thank you for the introduction. So my name is Anna Klucnika. I am a Latvian who grew up in the UK, which makes this meeting especially special for me. So thank you to the organizers for such a fantastic event. And I'm very lucky to present after a wonderful panel of speakers who have made my job a lot easier by introducing induced pluripotent stem cells and CRISPR-Cas9 gene editing, which is what we use at Laverock Therapeutics. We're a preclinical startup company based in England, just north of London. And we use gene editing induced gene silencing, so our technology called GEiGS, to develop new cell therapies for solid tumors. So as we've heard today, cell therapies are already in the clinic, and so this is the next generation of medicine is already being used. So we can use cell therapies in the form of autologous cell therapies, such as CAR T cells, where cells are taken from the patient. They are then engineered, expanded, and then put back into the patient. So for CAR T cell therapies, this is then used to engineer in a chimeric antigen receptor, which then recognizes and attacks the tumor cells. And when this is successful, this has huge patient benefit. But there's also a lot of challenges to cell therapies that are currently being faced. So there's can be off-target toxicity if in the case of CAR T cell therapies, there might be non-specific binding of the chimeric receptor. And also there's also a lot of CAR T cells can undergo exhaustion due to overstimulation, where there's activity of the cell therapy, not just in the tumor, but generally in the patient's body. And another problem with cell therapies is that often there aren't the resources, the time, or the immune cells available to perform the cell therapy treatments. And so instead, what's a very powerful and sort of promising way forward is sort of the use of allogenic cell therapies, where a donor is then used to harvest the cells, which are then engineered to make a master bank. But this is currently this is only possible with donor matching, because otherwise the donor cells will be rejected from the patient due to the immunogenicity. So we need to try and develop ways of generating hyperimmune cells that are invisible to the immune cells, so that the immune system, so that we can have master cell banks of cell therapies that can be used for wider patients. And so to be able to overcome all these challenges, we need new ways of engineering cells so that we can have stable, tunable, programmable, and specific gene expression changes. So if we consider the canonical technologies that are currently used for engineering gene expression, these allow us to do many fantastic things, but they also have some limitations. So if we wanted to generate a gene knockout, you would probably turn to CRISPR-Cas9, because you can generate a specific knockout of a protein using a single gene edit. You can check that this is specific to your target gene of interest, and this is stably inherited in the patient cells or the donor cells, because this is genetically encoded. However, as Pia mentioned, this is not programmable, so this is either on or off, the cells are mutated or not, and this is then in all of the cells. And it's also not tunable, so you can have a binary effect, so you can't have silencing of a protein or overexpression, so you can just generate a mutation. If you wanted to have more tunable gene expression changes, so to vary the level of the protein of interest, you could turn to traditional silencing methods, such as shRNA or siRNA, and this is where you can use a transient addition of siRNA, so small RNA molecules, to silence a protein, a gene of interest, or you can introduce a construct where these are then expressed in the cells, and this can lead to tunable expression, but this comes at a cost of being very unspecific, so it can lead to sort of lots of different off-target effects and off-target silencing. It's not stable because either the siRNAs are added transiently or the constructs are epigenetic silenced, and it's not programmable because you can only apply it to the cells or you don't. We can use more sort of advanced methods of CRISPR-I or CRISPR-A to try and achieve this in a different way. So then we can have tunable gene expression changes to increase or decrease the level of a target gene. And this can be stably inherited because we're engineering the cells. And we could do this programmably if using sort of quite simple synthetic gene expression circuitries to ensure that expression of constructs only occurs in a cell type of interest. But this comes at a cost of then having needing to edit large constructs into a cell to achieve this, this advanced sort of silencing. But what our technology, GEiGS, allows us to do is to achieve, with one single gene edit, specific silencing of a target gene that is tunable, so we can change the level of expression of the gene from, say, a complete knockout to 50% expression. And this is stably inherited because we do this by modifying endogenous genes, and we can have the silencing to be programmable, so context dependent, to give us more power to try and tackle different diseases. So how do we do this? So actually, just like how CRISPR-Cas9 uses the cell's own DNA repair mechanisms to achieve DNA editing, our silencing technology uses the cell's own silencing mechanisms to achieve gene expression changes. So eukaryotic cells are already equipped for programmable gene silencing. So our genomes encode 20,000 protein encoding genes, which are transcribed into mRNA transcripts, which are then translated into the proteins. And so gene expression can be modulated by either affecting transcription or translation. And there are around 2,500 microRNA genes that are also encoded in the human genome that are expressed and processed to form mature microRNAs that are complementary to the target mRNA sequence. And this then results in reduced protein synthesis by either resulting in reduced translation or in degradation of the mRNA transcript. And actually, it's very topical because just this week, the Nobel Prize for Physiology or Medicine was awarded to Ambros and Riekstina for microRNA, the discovery of microRNAs and their role in post-transcriptional gene regulation. So what we have utilized this for is for gene editing and gene silencing, so GEiGS. So we use conventional gene editing technologies, such as CRISPR-Cas9, to retarget endogenous microRNA genes by changing a few base pairs so that when the microRNA gene is expressed, it instead targets our gene of interest to result in protein silencing. So this is a way that we can achieve specific, tunable, programmable, and gene expression changes without actually interfering with any protein coding genes. So here we have engineered iPSC cells, so induced pluripotent stem cells, so that a GEiGS engineered iPSC where we have redirected a microRNA to silence a target of interest, in this case beta-2M. And this shows really stable silencing of the gene. So in light gray, we have the beta-2M knockout cell where there's no expression of the target protein. In black, we have the wild type where there's sort of normal expression of beta-2M. And in red, we have one GEiGS design where there's partial silencing of beta-2M to around 90% of normal gene expression. And in blue, we have a different GEiGS design where we have over just 10% of normal expression is remaining. So we have very sort of homogeneous, very tight expression profiles, and depending on the design, we can achieve different levels of silencing. And what's really nice is we can also achieve a multiplex gene silencing with just a single edit. So here we have engineered iPSC cells to silence both two targets, both beta-2M and GFP. In black, we have the wild-type cells with normal expression of GFP and beta-2M. And then we have engineered a microRNA to target these two proteins. And in blue, we have a heterozygous iPSC cell that shows silencing of both GFP and beta-2M. And in red, we have a homozygous knock-in where there's two copies of our engineered microRNA, and so the silencing is even better. So we have lower expression of both GFP and beta-2M in our homozygous GEiGS cells. And what I think is the most coolest feature of GEiGS is that we can have context-specific silencing. So depending on the microRNA we just choose to engineer, we can then tailor the expression of the GEiGS engineered microRNA and therefore have context-dependent gene silencing. So in this case, we've engineered iPSCs to generate cell lines that have a cell-type specific microRNA redirected to silence beta-2M. The engineered microRNA of interest is strongly expressed in macrophages, but not in induced pluripotent stem cells. So we've confirmed this by next-generation sequencing. And then we use CRISPR-Cas9 to engineer the iPSC cells so that this microRNA of interest is redirected to silence beta-2M. And in the iPSCs, there's normal expression of beta-2M in both the control, where the microRNA is simply knocked out, and in the GEiGS line in red, where we have redirected the silencing of our microRNA of interest. So there's normal silencing in iPSCs, but when we differentiate the cells to either the monocytes and then to macrophages, the scaffold microRNAs is switched on, and therefore the target is then silenced. So we see programmable, context-specific silencing of our target protein beta-2M. And another feature of GEiGS is that this silencing can be achieved without minimal off-target effects. So we performed transcriptional analysis of our engineered clonal GEiGS iPSC lines, and we didn't find any detectable loss or gain of function expression changes in our mRNA transcriptome. So on the left here, we have a beta-2M knockout cell line where there is only beta-2M expression is significantly downregulated as expected because we've knocked out the gene. In the middle, we have a microRNA control line where we've disrupted the microRNA that we're engineering. And again, we don't see any changes in gene expression. And then in our GEiGS line, we only see significant changes in our target gene beta-2M, because this is the one that we have intended to silence, and we don't see any off-target effects. So in summary, our unique technology called gene editing induced gene silencing, or GEiGS, leverages endogenous microRNAs that are known to regulate gene expression. And we use minimal gene editing to recode the microRNAs to selectively and conditionally silence gene targets of choice. And this has really great advantages because it's stable and physiological, because we're using endogenous microRNAs that are expressed at physiological levels, so we don't get this off-target silencing that you see with other traditional methods. It's tunable and multiplexable, so we can tune the level of silencing depending on the design we use and the final product that we're looking for. And it's also multiplexable, so we can target multiple targets at the same time, which means that we can develop more complicated products. And it's programmable, so the gene silencing is linked to context-specific microRNA expression patterns so that we can achieve gene modulation in the cell type or the cell state of choice. So now we've developed a flexible and efficient pipeline for screening therapeutic GEiGS iPSC-derived cells to go from therapeutic strategy to differentiating cells into a therapeutic product for cell therapies. And our main focus at the moment is developing immune cell therapy for solid tumors. So we start off with the goal in mind, with the therapeutic strategy, and identify our target genes that we wish to modulate the gene expression for. We perform transcriptional profiling to identify microRNAs that are expressed specifically in the target product that we're trying to develop. And then we have a computational pipeline that takes in the target gene, the context-specific microRNAs, and then does iterative designs to output thousands of GEiGS microRNA designs that are redirecting the endogenous microRNAs to silence our new target. And so there's different ways that we can perform this design screening to try and narrow down the thousands of different designs into a specific number that we can then use for cell line engineering. So we can perform arrayed screens where we overexpress the GEiGS design one by one in different cells. And so here we have tested 14 different GEiGS designs compared to the control wild-type cells. And we see that each of these 14 different designs leads to different levels of silencing of our target beta-2M. And so we can then pick out which design we want to use. Say we want to get a strong amount of silencing, we want to only have 10% of the protein left over, or if we only want to have minimal changes, then we can choose a different design. And we've also developed pool screening approaches where we can screen over 6,000 different designs at the same time to then enrich for the ones that show efficient target silencing to then narrow down this search and screen for many more targets and many more designs in the same go. So then once we have identified the GEiGS designs that we wish to use, we then proceed into cell line engineering. So we utilize CRISPR-Cas9 at the moment to achieve the cell line engineering. Is it clicking? I'm trying to click, but it's not working. Okay, great. So we have a streamlined workflow for generating iPSC-engineered GEiGS lines using CRISPR-Cas9 HDR editing. So the first step in this process is guide RNA screening and HDR donor design. So that's really nicely outlined by the previous speakers. So we've optimized this, and so we know that we need to test at least 3 different guides per microRNA to find the most efficient guide RNA. As we were shown earlier, sort of different targets can lead to different editing efficiencies. And so to overcome this, we try and screen as many different guides as possible. And we use short 200 base pair single-stranded DNA templates to try and achieve efficient iPSC HDR knock-in efficiency. And we also tend to test both the target and non-target strand to try and, again, achieve optimal editing efficiency for different targets that show different levels of efficiency. And then iPSCs show quite low levels of gene expression changes. So we then need to, iPSCs tend to have quite low editing efficiencies, so we use small molecule inhibitors of non-homogeneous end joining and also use cold shock to try and boost the HDR efficiency. But even then, we say we only have between 1 and 10% of our cells that we've engineered have the edit of interest. So we use long-read amplicon sequencing using Oxford Nanopore technologies and have a custom analysis pipeline to quantify the efficiency of gene editing. Once we have identified a pool of interest, we then use single-cell cloning to identify single clones. And we have optimized the single-cell seeding and recovery method and have also implemented automated liquid handling and high-throughput genotyping of the clones, again using long-read sequencing to try and screen through as many clones as possible. So by implementing this index genotyping, where we culture cells in 96-well plates and add index primers to then pool some samples from the same plate, we're able to decrease the time that each workflow takes from about 10 to about 7 weeks, and then increasing automated liquid handling also allows us to further increase the speed of the workflows, kind of cutting down the time, turnaround time around half, which allows us to scale up the gene editing processes so that we can generate different cell therapeutic cell lines. So once we have identified and isolated our GEiGS edited iPSC line, it's very important to perform quality control checks before using these for any further studies. So we perform QC checks, including pluripotency, genomic stability, growth tests, sterility testing, to make sure that we have high-quality engineered lines. Once we have these iPSC lines, we then use them to differentiate them into a therapeutic cell product. And currently, we're working on 3 main programs. So one of the programs that we're working on is to generate hypoimmune cell therapies. So what we are trying to do is generate pancreatic islet cells that have hypoimmune status. So we generate GEiGS-engineered clonal iPSC cell lines, which we then differentiate into pancreatic islet cells. And so we've specifically engineered them so that the GEiGS expression is specific to the pancreatic islet cells and results in silencing of MHC1 expression so that the cells can avoid immune rejection and so can be transplanted into patients to result in insulin independence without any need for immunosuppression. We're also working on solid tumor therapies. So we are working on improving T cells. So we're using GEiGS to enhance CAR T cell therapy by having tumor microenvironment-induced checkpoint inhibition and also generating hypoimmunogenic allogenic immunotherapies using CAR T. So this will be generating T cells programmed for enhanced potency and resistance to T cell exhaustion. And the third program that we're working on is generating macrophages that have increased specificity and activity against solid tumors. So normally macrophages, when they enter the tumor microenvironment, they become a tumor-associated macrophage that is associated with increased tumor persistence. But we can engineer these macrophages so that when they are in the tumor microenvironment, instead of becoming tumor-associated macrophages, we initiate an anti-tumor activity to help target solid tumors. And so in this way, we're generating GEiGS macrophages programmed for anti-tumor efficacy and resisting tumor-associated macrophage formation. So in summary, click through. I've shown you today how we're using GEiGS to reprogram the cell's own control systems to develop advanced therapies that respond to specific cellular contexts or disease states. Thank you very much for listening, and I'd like to take any questions or contact me later on in the day. I'm always happy to connect. Thank you very much.
Any immediate questions?
Yeah, I would like to invite all speakers to the panel on the stage. Take a seat on the stage, and then the audience will be invited to ask questions. So, yeah, I hope there is some help with microphone. Yeah, questions, please. Questions, welcome. Those are really fascinating talks. Maybe I can start with a question to Anna, because we are also doing some research on tumor-associated macrophages, and actually what are the features that keep these macrophages like this anti-tumor, with this anti-tumor function?
Yeah, that's That's a great question. So we're screening different targets to try and identify the best targets to silence in order to achieve the efficient gene silencing. So there's different targets that we're looking at that have been looked at in the literature, but we're also looking forward to performing screens to identify new targets. So in order to generate the screens, we're using CRISPR-Cas9 to knock out or silence different targets and then see whether they get polarized or not. So the specific targets, unfortunately, I can't release too much about them, but maybe we can talk about it afterwards.
Okay, I understood your strategy. Yeah, then thank you so much. Yes, maybe there is a question from audience.
So actually,
yeah. Okay. Yes, please go ahead.
Also for Anna. So maybe I missed it, but you said you talked about like screening for activity of mRNA, but do you also screen for specificity?
Yeah, that's a great question because we're always sort of conscious that we want to try and have a specific gene silencing in both the cell state and according to different gene products. And I guess we have two different challenges where we need to look for specific changes. So first, we need to check that our editing approach hasn't led to any off-target effects. So we try and choose the guide RNAs with the sort of lowest off-target profile. And we're looking at different ways of trying to screen for the off-target approaches. So this is something that I'd be really interested to hear the panel's thoughts on this, because there's sort of very many different approaches to screening for guide RNA off-target sites. And we could try and perform sort of PCR and Sanger sequencing or NGS of predicted sites. But since we're generating clonal sites, we could also perform whole genome sequencing. I think when we try and use these for clinical trials, we'll definitely have to do that and sort of have control lines to do that. And then once we have isolated a line that we're interested in, then we need to make sure that our microRNA isn't having any off-target silencing effects. So one way we can do that is by whole transcriptomics. So that's the current way that we're doing it, sort of to really see the robust functional effects of it.
And can you also do some prediction of specificity of microRNA?
Yeah, definitely. So we can, the computational pipeline sort of can look at where it's predicted to bind and try and minimize that. So we're trying to achieve that, yeah. And so what we've currently seen is there's very little off-target effects, but I think it will depend on the design, which microRNA we're modulating, because it might not just be the sequence that we're introducing, but the microRNA that we're editing might also be essential, so then we have to be conscious of that.
I had actually another question to Anna as well, because I was very impressed of this work, and I can see how I could use this in my research as well. So I was curious about the type 1 diabetes project. Have you characterized how well these, let's say, pancreatic islets become like functionally? Like, have you done like single cell sequencing or that kind of characterization to see are they really as functional?
Yeah, no, I think we haven't gone to that stage yet, but there's lots of other companies that are really, really far along with developing the pancreatic cell therapies. So it's really exciting. So you can generate pancreatic organoids and test for insulin release. And so now there's different clinical trials going on where they're implanting the pancreatic islets into different patients. So I think it's really important then to make sure that you have that functional validation.
Thank you.
Yeah, if I may ask a question to Pia, a layman's question. How do you make CRISPR-Cas enter iPS cells?
Right, thank you. Like that? Yeah, so we use nucleofection. So we use RMP and the sort of most high fidelity Cas9 that we can find. And that works really well with iPSC. So we get, well, let's say that it works reasonably well, then we have like maybe 80% editing efficiency. And we also think that using RMPs and high fidelity Cas9 really reduces the off targets as well. So that's how we introduce it.
Thanks. Yeah, and also I was very fascinated by talk given by Giedrius. So you have discovered this special Cas12 and you named it after Lithuania. And has it been now used for some, like into some disease, possible disease treatment approach, or is it still like done more like a preclinical or basic research?
So at this point, we're doing on like improvements of the tools, but we're starting to collaborate with other companies who will lead to clinical translation, or also, as I mentioned, another also field is agro, which is also very interesting to us.
Thank you. I sometimes seen places in the city that they proudly put on the door like non-GMO, like cafes and things, but I hope this will change. Okay, maybe there is meanwhile a question. Yes, please.
Yes, I addressed that to Anna, but maybe also you have you wonderfully described, for what I understood, that you have a therapeutic strategy until the end. So if somebody comes with an idea, how much time does it take at the end of not of the day, of course, but what does it mean?
Yeah, that's a great question, because this is something that is really that we're asked a lot, because the technology is so useful in many different applications. And so what we're trying to do is narrow down that timeline. But let's say you can, target validation can take many number of years, but once you have a target, we can then generate the different designs in a couple of months. And with the pool screening approach to test which design is the best, that can take another, let's say, 4 months. So in 6 months' time, you can go from having a target of interest to having your GEiGS designs that are efficient at silencing. And then it will take another sort of maybe 2 months to 3 months to generate a, if you're using iPSCs, to generate a clonal iPSC line and validate that, perform all the QC checks on it. So say in approximately less than a year or so, you can then include differentiation of that iPSC into the final cell product. So you can have, in total, about a year to go from target to have a differentiated cell product that you can then use for in vitro validation or in vivo testing or for clinical trials. And I think as we develop more things further, we can try and shrink that timeline more.
Okay, thank you. I think we have approached the end of our session, so I would like to thank the wonderful speakers once again. And thank you for your attention, and thanks. The session is closed now.